Okay, Assalamu alaikum, welcome back. We are now ready to begin examining the quantitative aspects of how truly special the genetic code is. And you will see that there is an amazing level of optimality. The first substantive paper I am aware of that tried to look at these sorts of questions was by David Haig and Lawrence Hurst, published in the Journal of Molecular Evolution in 1991. So you can see that we're pretty much on the cutting edge of science now. We're doing stuff that, began to be understood late in the 20th century. And when they looked at the error minimization of the genetic code in terms of its ability to resist point mutations, they found that the standard code is better than all but 0.02% of random codes, so that it is better than 1 in 5,000 random codes. How do we explain that? Well, we explain it by the redundancy we've talked about before, that if you change one of the codons, you often will not change the amino acid because multiple codons code for the same amino acid. And the question becomes, if I want to look at the structure of proteins with polar amino acids that have charge and amino acids that are not charged and things that would affect the ultimate structure of proteins if a mistake happened, the first analysis suggested that the standard genetic code was extremely efficient, better than 0.02% of randomly generated codes, but this was looking just at point mutations, single base pair changes. Some years later, the team refined their analysis. Once again, Professor Hurst, now working with who I believe was his student at the time, Dr. Freeland, took another look at the optimality of the genetic code, this time making the simulation a little bit closer to nature. Remember, all of these are mathematical simulations. One of the things they did, for example, was now take into account that not all point mutations are equally likely. The initial paper that we looked at assumed that all of the point mutations were equally probable. But if we look at nature, we see that a mutation of A to G is more likely than A to C or A to T, for example. And when this situation was taken into account, it turned out that the standard genetic code, our genetic code, is even more efficient and more optimal than the first analysis showed. And in fact, that is the title of the paper. It now turned out that the genetic code is 1 in a million. That is its level of optimality or efficiency. Now let's take a look at a little bit more detail. And remember Darwin's quote at the beginning, that when he looks at the details, he doesn't see evidence of design. Well, we are saying precisely the opposite, that the more we look into the details, the more the evidence of design looms as the best possibility. We have discussed point mutations. Now it's time to broaden our scope and state that those are not the only sorts of mutations in DNA. Another very important sort of mutation is called the frameshift mutation. Remember, the DNA is a long string of bases made up of the 4 letters, and they are read off in 3-letter groups known as codons, each codon coding for an amino acid. We talked about stop codons that tell the protein to stop, and there's also a place to start. But what if we shift over by 1 base pair, by 1 letter, and start in the wrong place? That's known as a frameshift mutation. So if you take a look at this diagram, we see what a normal translation would be. For example, we begin with the three-letter codon ACG, and then we go to AGG, and ACG is for threonine and AGG is for arginine, and that would be the order of the amino acids in the protein. But what if we make a mistake and shift over by one letter and then start taking triplets? Well, you see then that the amino acids get totally messed up. They're completely different amino acids. Each group of three still codes for an amino acid, but now we're coding for the wrong amino acids than we intended. And you see a diagram for a +1 frame shift or a -1 frame shift. But let's take an English example. If we have the sentence made up of three-letter words, the cat ate the rat who was sad, and we drop the first T and we start taking three-letter groups from the H, we now see that these three-letter words get completely messed up. A couple of them end up still being words, but the entire meaning of the sentence gets changed. Now, with DNA and RNA and amino acids, things are a little bit different because each three-letter word makes sense, but you can see that the order of the amino acids gets entirely changed. So the DNA is not only prone to point mutations, it can also undergo frameshift mutations. And the question becomes, how good is the standard genetic code at resisting frameshift mutations? That if a frameshift mutation occurs, how likely am I to still get the same protein or a very close protein that can still be functional, fold in the same way, and so forth? And how does this standard code that we have compare to the other possible codes that could have been? So here I'm showing you a paper that undertook this analysis. And look, this paper is from May 2018, so you can see that this is really now the cutting edge of science. And it looked at the ability of the standard genetic code to resist point mutations, frameshift mutations, translational errors, and compared the standard genetic code to 1 million randomly selected codes. Remember, we said that depending on how you choose to reassign the codes, you can have anything from several million possibilities to 10 to the 80 or 10 to the 84th, billion possibilities. But in any case, if you take a lot of these random genetic codes and analyze them for efficiency, and analyze how good they are at resisting, or not really resisting, but correcting for, being unaffected by point mutations, frameshift mutations, and so forth, we see a very interesting result. So here's a typical graph from this paper, and let me explain to you in rough terms what this graph shows. It is looking at different types of possible errors or mutations, and it looks at the natural genetic code versus 1 million randomly selected codes, which could have been our genetic code, and looks at sort of some measure of the error or the cost of these mutations on the code. And if you look at the 1 million different randomly selected codes, you see that they form a distribution, sort of a bell curve. And for each of these possible sorts of errors that could creep into DNA, you see that the natural genetic code, which all living organisms use, is outside that distribution. It's to the left of the distribution, meaning that with extremely high statistical significance, the natural code is essentially better than any randomly selected code for each and every type of possible error. And this is an amazing result. And the authors summarize these results here, and let us go over them together. And once again, I hope you're not put off by this level of detail. If we want to do science and we want to understand how special things are, we have to dig deep. And so they say, but our results along the lines of lots of other work, like Freeland, we've already looked at the paper by Freeland and Hurst, show that there's a clear non-random structure underlying the standard genetic code, the SGC, making it remarkably robust not only against point, but also against frameshift mutations. And they say the results show that the SGC, they talk a little bit about how efficient it is, and that it outperforms more than 99.99% of 1 million randomly generated codes. Now here's the important part, the last sentence. This effect even got stronger for the combination of all three proposed measures, indicating that all three factors might have been contributed independently to the evolution of this sophisticated, robust, and universal coding. What does that sentence mean? It means that when we look at each of the three possible errors, point mutations, frameshift mutations, translational errors. We haven't gone over translational errors because this is, again, just to give you a feel for things. The genetic code, the standard genetic code, is better than essentially all other codes. But when you look at the combination of all three, it becomes even better. It's remarkable. That's like, for example, saying, I have a person who's better than almost everybody in math. And I have a person who is better than almost everybody in sports. And I have a person who's almost better than everybody in literature. Those are three very separate things. Now, it would be quite remarkable if that person was the same person, if that person was the best in music, in sports, and in literature. That makes it even more remarkable if you're 1 in a million in sports and someone is 1 in a million in math and someone is 1 in a million in literature. Well, how likely is it that someone is the exact same person each and every time? That makes that person even more remarkable. And that's what this paper is saying about the genetic code, that it is amazing in each of the measures separately. But when you take the combination of all three measures together, it turns out that this code is an amazingly efficient, a remarkable code. So what happens when the analysis is refined still further? So we've already looked at a couple of different refinements of the analysis, taking into account the frequency of point mutations, that A to G is more common than A to C or A to T, for example, and that would be the equivalent of taking into account when we discussed our simple 3-bit correction, that it is not as likely for a 1 to go to a 0 as a 0 to go to a 1 when we're transmitting bits. You could see that that would make a big difference in the math. Then the analysis was further refined by looking at not just point mutations, but frameshift mutations as well. Now the analysis is refined even further by another group, looking at different amino acid frequencies in proteins. So far the analyses, which are mathematical models, have assumed that the amino acids are equally likely, and any mistake in an amino acid is counted against the code equally. But if you look at nature, if you look at our chemical structure, there are certain amino acids that are more common than other amino acids in our makeup. An easy way to understand this is if you look at the English language, the letter E is much more common than the letter Z in the words that we use. So now an analysis takes into account these different amino acid frequencies in proteins. And you could see, for example, that if there is a sort of error that would affect the letter E, that would much more significantly impact English than an error that would affect the letter Z. So when you do that same sort of analysis on amino acids and not on English letters and words, it turns out that the genetic code, the standard genetic code, our genetic code, is even more amazing than all of the previous analyses had suggested. I've put up a blurb of the results, and you're certainly free to pause the video and read it in more detail. But the bottom line, which you can see in the last part here of the results, is that when this measure is taken into account, it is seen that the genetic code is around 2 in a billion. So it's not 1 in 10,000, it's not 1 in a million, it's 2 in a billion. And again, if you now put this together with the other measures that we've already talked about, it means that this code is an absolutely phenomenal, remarkable code. And things don't just stop there. When we take a look at DNA, so far we've been talking about the DNA carrying the information to code for proteins. It turns out, however, that a lot of the DNA in our bodies doesn't code for proteins. It does other things, regulatory functions and so forth. There's additional information. Because if you look, for example, your brain cells, your heart cells, your liver cells all contain the same chromosomes, the same DNA. Well, then what makes one cell a brain cell? What makes one cell a heart cell? What makes one cell a liver cell? Well, it is regulation. Which genes are expressed and how much? That is how one cell becomes different than another cell, because they all carry the same stock of DNA, because they all have the same chromosomes. So a lot of the DNA contains additional information, regulatory information. So when we're going to look at the efficiency of DNA, of the standard genetic code, we don't just want to look at how will proteins be affected by the various types of mutations. We also want to look at how this additional information that is also coded and stored in the DNA, the regulatory information, would be affected by mutations. And it turns out that our standard genetic code is also, this paper says, nearly optimal for allowing additional information to be stored within the protein coding sequences. And so that's something we may never even have thought to look at or to analyze. Luckily, this group from the Weizmann Institute did do that, and this is their result.